Welcome back guys. Here we are going to continue our lecture of CET Physiology recall based question. So in this part we are going to discuss all these topics. Again remember these are recall based questions. So there may be little bit changes in a question, but remember guys, the topic remains same. Okay. So now starting with a question, arrange the order of sequence of events after P wave in ECG. So first we will talk about cardiac cycle. What is cardiac cycle? It is sequence of events of what? Electrical and mechanical. That is, sequence of electrical and mechanical events is nothing but your cardiac cycle, which repeats with every heartbeat, and it is well understood with the help of Vickers diagram. So now let's have a look at Vickers diagram. Okay, here you can see this is ECG, okay, ECG wave. Here you are having a pressure column where this one is your atrial pressure wave, this one is your ventricular pressure wave, and this one is showing your ventricular volume, and again over here heart sound is there. We know cardiac cycle starts with what? When SA node fires. SA node fires, it will generate P wave on ECG. Okay, you will get P wave on ECG. What will happen after that? After that, you will get atrial contraction. What will happen to this atrial wave? In atrial contraction, there will be increase in atrial pressure. So, you will get A wave on what? JVP, yeah, because we know that the atrial pressure is reflected in the JVP waves. So, this one is your A wave. What is happening to the ventricular pressure? Ventricular pressure is increasing during the atrial contraction. What is happening to the ventricular volume? Again, it is increasing. Now, after the atrial contraction, after the P wave on ECG, there is QRS complex, and after QRS complex, you get isovolumetric contraction. What is happening in isovolumetric contraction? After the P wave, that is after the atrial contraction, now this wall, AV wall gets closed, and this closure of the AV wall is the beginning of your isovolumetric contraction. Again, this closure of AV wall, it produces first heart sound. Write it down, first heart sound. It is seen in the beginning of isovolumetric contraction. Now, walls are closed, both walls, AV walls and semilunar walls. They both are now closed. Now, ventricles are contracting. So, here you can see that ventricular pressure is now here increasing. What about ventricular volume? As both the walls are now closed, so ventricular volume remains constant. What is happening to the atrial pressure? Remember guys, during this isovolumetric contraction, there is bulging of AV walls into the atrium, which is causing increase in atrial pressure and which is reflected in the JVP as C wave. Okay, C wave is due to bulging of tricuspid wall into the atrium. Got it? So, we are having C wave on JVP. What is the next phase after the isovolumetric contraction? There is ventricular ejection. It means ventricular ejection, it starts when semilunar walls get open. Now, blood is ejected from the ventricle into the aorta and into the pulmonary trunk. Now, here you can see the ventricular pressure is increasing. And what is happening to the ventricular volume? Ventricular volume is decreasing. And what about atrial pressure then? Remember guys, during the ventricular ejection, this AV wall gets pulled inside the ventricle. What will happen due to that? Due to that, there will be decrease in atrial pressure, which is seen as X descent on JVP. Okay, X descent, it is seen during the ventricular ejection. And remember guys, during this phase, you will get P wave on ECG. What is the next phase? Next phase is your isovolumetric relaxation. Isovolumetric relaxation, it starts with closure of semilunar wall. Write it down, isovolumetric relaxation, it starts with closure of semilunar walls. And this closure of semilunar walls, it produces second heart sound, S2 sound. Okay, and now as walls are closed, both semilunar wall and AV walls are now closed, and now ventricles are relaxing. So, what is happening to the ventricular pressure? Ventricular pressure is decreasing. What is happening to the ventricular volume? As both walls are closed, so ventricular volume remains constant. Now, what is happening to the atrial pressure? Remember guys, during the isovolumetric relaxation, this atrium is getting blood from the IVC, SVC and also from the pulmonary veins. It means pressure in the atrium is increasing, which is seen as V wave on JVP. Got it? So it means V wave you will get in isovolumetric relaxation. Now what is the next phase? Next phase is your ventricular filling. It starts when your AV walls get open. So now AV walls get open and now blood is coming from the atria into the ventricle, leading to what? Ventricular filling. And remember guys, here ventricles is very relaxed, so its pressure is decreasing. Ventricular pressure is decreasing. And what is happening to the ventricular volume? Ventricular volume is increasing. What is happening to the atrial pressure? Remember guys, here you can see that the blood is continuously coming from the atrium to the ventricle. That's why here atrial pressure is decreasing, which is seen as wide descent on JVP. Got it? So, in nutshell, we can say that first you will get the P wave followed by A wave followed by first heart sound, then you will get T wave, then you will get the ventricular filling. Got it guys? Okay. So, now that's why the answer to this question is B. Now, next question. Along the length of the proximal tubule, concentration of which of the following substance increased? First, we will talk about substances in the proximal tubule. Remember guys, in the early part of the PCT, in early proximal tubule, sodium preferentially carries glucose, amino acid and bicarbonate. Okay, sodium prefers glucose, amino acids and bicarbonate in early part of PCT. What about chloride? Remember, chloride concentration, it increases as water is also reabsorbed from the early part of the PCT. Then what about the distal part of PCT? Remember guys, sodium is reabsorbed with chloride in this distal part of PCT. Got it? And one more thing you should know, that sodium concentration and osmolarity, it remains constant throughout the PCT. Got it? These things you should know, that from the early proximal tubule, glucose, amino acid and bicarbonate, they get reabsorbed along with the sodium. So, you can say that the chloride concentration in PCT, in early part of PCT is increasing. Okay? And this chloride gets reabsorbed from the distal part of PCT. Okay, that's why in this diagram you can see that if this is the showing your sodium and osmolarity, I told you it remains constant. Okay, and here you can see the bicarbonate, glucose and amino acid level in the PCT, it is decreasing, while you can see the chloride, urea and creatine level is increasing. Got it? That's why the answer to this question is chloride, whose concentration is increasing. Now, arrange the following based on filtrability in glomerulus. So, first we will talk about the dextran, then we will come back to this question. So, what is actually dextran? Remember guys, dextrans, they are polysaccharides which is used to measure glomerular permeability. Yeah, for checking the glomerular permeability, we are using the dextrans. Got it? Again, you should know that this dextran show that electrical charge affects the filtration. Okay, so these dextrans, it just show that whether the electrical charges affect the filtration or not. So here, one more thing you should know, that for any given molecular radius, okay, for any given molecular radius, positively charged molecule, they are filtered more than the negatively charged molecule. Write it down, that positively charged molecules, they are filtered more than the negatively charged molecule. While if you see the neutral molecules, then write it down that neutral molecules, they are filtered more than the negatively charged molecule. Got it? So, this thing can be represented with the help of the graph. So, here you can see that polycationic dextran, okay, polycationic dextran is filtered more as compared to the neutral dextran, and neutral dextran is filtered more as compared to the polyanionic dextran. Got it? Write it down. So now, why there is difference in permeability? Remember guys that this negatively charged basement membrane and podocyte, they are having a negative charge which restrict the large negatively charged molecules including plasma proteins. Okay, this is the reason for difference in filtrability across the glomerular membrane. Okay. So again, I have shown you this graph that polycationic dextran filtered more as compared to the neutral, neutral is filtered more as compared to the polyanionic dextran. That's why the answer to this question is B. Got it? Now next question, match the following. After hyperpolarization, depolarization, repolarization and channel's name are given. Okay, first we will talk about action potential. This is action potential of a nerve. Okay, here this is RMP. RMP of nerve is minus 70 millivolt. Okay, this is your depolarization, this is your repolarization, and this is your after hyperpolarization. Okay, we know that depolarization is due to opening of voltage gated sodium channels. Due to opening of this channel, we are getting influx of sodium, and this repolarization is due to opening of voltage gated potassium channels, which are doing what? Which are doing efflux of potassium, causing repolarization. And what about this after hyperpolarization? Remember guys that this after hyperpolarization is again due to voltage gated potassium channels. Why? Because these voltage gated potassium channels, they are slow to open and slow to close. So, due to slow closure of voltage gated potassium channels, you get after hyperpolarization in which membrane potential goes below the level of resting membrane potential. Okay. So, that's why the answer to this question is B. Which of the following statement about the excitation contraction coupling in skeletal muscle is correct? So, first we will talk about excitation contraction coupling in skeletal muscle. We know that excitation contraction coupling, it starts when alpha motor neuron discharges. Alpha motor neuron discharge, action potential is produced. Now this action potential, it travels along the T tubule and it reaches where? It reaches near the sarcoplasmic reticulum and it causes release of calcium from the sarcoplasmic reticulum, and now this calcium binds with troponin tropomyosin complex and we get muscle contraction. Okay. So, here action potential is produced, it travels along the T tubule, it causes release of calcium from the sarcoplasmic reticulum, not mitochondria, from the sarcoplasmic reticulum. Now, this calcium binds with troponin tropomyosin complex leading to muscle contraction. Okay. So, one more thing you should remember guys for this is, if this is your T tubule, then remember in case of skeletal muscle, in the T tubule you are having DHPR receptor. Okay, and if this is a sarcoplasmic reticulum of a skeletal muscle, then you are having RYR receptor in the sarcoplasmic reticulum. And remember these two, DHPR and RYR, they are attached to each other mechanically or physically. What does it mean? It means whenever action potential comes, there is activation of this DHPR channel. Whenever this DHPR gets activated, it just pulls the RYR from the sarcoplasmic reticulum. And remember, whenever RYR gets pulled out of the sarcoplasmic reticulum, it causes release of calcium from the sarcoplasmic reticulum into the sarcoplasm of a skeletal muscle, leading to contraction. Okay guys, you should know this point. So, that's why the answer to this question is 1 and 3. That is, extracellular calcium plays no role. It is a sarcoplasmic reticulum calcium which plays a role in excitation contraction coupling of skeletal muscle. Okay. And what about intracellular source of calcium is mitochondria? No, it is sarcoplasmic reticulum. So, it cannot be the answer. What about this electrical event is followed by mechanical event? Yeah, first action potential is produced, then you will get the action potential. But one more thing you should remember, that extracellular calcium plays no role in case of excitation contraction coupling in skeletal muscle. But what about smooth muscle and cardiac muscle? Remember guys, for smooth muscle and cardiac muscle, extracellular calcium is important for excitation contraction coupling. Okay. Now next question, diarrhea in Vibrio cholera is due to disruption of. Remember guys, the answer to this question is zona occludens. Let's see how. Remember, zona occludens is nothing but your tight junction. Okay, your tight junction is also called zona occludens, and this tight junction, that is zona occludens, it's like a zip. Okay, it is like a zip which fuses the apical margins. Okay, apical margins of the adjacent epithelial cells together. That's why this tight junction, it regulates paracellular movement. Okay, it is very important for regulation of paracellular movement. Why? Because it is fusing the apical margins of adjacent epithelial cells. So, remember one more thing is that the proteins which are forming this tight junction, write it down, it is very important. Claudin, occludin and JAM. What is JAM? It is junctional adhesion molecules. These proteins are there in your tight junctions. Then sites. Sites are blood-brain barrier, renal tubules, intestinal mucosa, gastric mucosa, urinary bladder mucosa, Sertoli cells of testis. Guys, these sites are very important. Okay, these are the sites where you will get the tight junction. Then, how Vibrio cholera is causing diarrhea by affecting the, by what? By disrupting zona occludens, let's say. Vibrio cholera, remember guys, it is producing zona occludens toxin which is destroying the tight junction, leading to opening of intercellular tight junction, which will increase permeability of the small intestinal mucosa, leading to diarrhea. Okay. That's why the answer to this question is zona occludens. Diarrhea in Vibrio cholera is due to disruption of zona occludens. So, in this part of video, we have covered all these topics which were asked in INI CET exam. Thank you very much for watching this video.